Hello again, Dr. Summer Smith. Once again, my name is Catherine Graves. Today is May 31st, 2019. Today I'm going to be giving a presentation on determination of sex or gender using genes or versus hormones. So before I get into determination of genes versus determination of sex versus genes and hormones, I want to give a little brief background of the sex organs. So males and females, we have sex organs that consist of three different structures. We have first the gonads. The gonads are the organs that produce gametes, so produce either eggs and sperm. So male gonads are the testes and the female gonads are the ovaries. And then our second structure of sex organs is the internal genitalia. Their internal genitalia is the accessory glands and ducts that connects to the outside environment. And then finally we have the external genitalia, which is all the external reproductive structures that are present on a male or versus a female for their sex organs. So going into the genetic factors that determine sex, so every individual has 46 chromosomes, so that's 23 pairs, and we also have one pair of a sex chromosome that is called our gonosome. So the female has two X chromosomes, and the male has one X chromosome and one Y chromosome, and the presence of the Y chromosome is the determination of the male sex. And the mother gives offspring two chromosomes and father gives one X and one Y. And if the individual in the early development has an X and a Y chromosome, that Y chromosome is going to determine that they're a male. So here is a diagram of the egg-producing cell, and we have the sperm-producing cell on the right-hand side. So during meiosis, the egg-producing cells split into maternal gametes, which are the eggs, into one X chromosome and one X chromosome. And then when the sperm-producing cell undergoes meiosis, we have one X chromosome and Y chromosome. And like I said before, the Y chromosome is a paternal gamete, which is a sperm. So when fertilization happens, we have crossing over, and then we have the fertilized eggs that could be either XX, XX, XY, or XY. And like I said before, if we have the XY chromosome, that individual is a male. So influence of hormones on gender in early development. So before differentiation occurs and an individual inside the embryo is determined to be either a male or female, we have embryonic tissues, and the embryonic tissues are bipotential because they can either develop into a male or a female before differentiation occurs. So bipotential internal genitalia consists of two pairs of accessory ducts, which eventually develop into the male or female reproductive structures later on in the development of the embryo. So the first is the Wolffian duct, which is derived from embryonic kidney cell, and then we have the Müllerian duct. So a continuation of this topic of the influence of hormones on gender in early development. So the sex determination depends on the SRY gene. If the individual is male, then the gonadal cortex regresses, and the gonadal medulla forms a testes, which is a Wolffian duct forms epididymitis, the vas deferens, the seminal vesicle, and then the Müllerian duct regresses. If it is a female, then the gonadal cortex forms an ovary. The gonadal medulla regresses, and the Wolffian duct regresses, and the Müllerian duct becomes the female sex organs. So as you can see, we have two different branches of development depending if you're a male or you're female. So here is a good diagram that shows the sexual development in the human embryo. So on the left side we have the Müllerian duct, and we also have the Wolffian duct, and both of these at this time are bipotential gonads because they could either develop into a female or male sex reproductive system. And in the female, after about 6 weeks, the internal reproductive organs have the potential to develop into either these female or male structures. So first, the gonadal cortex becomes the ovary in the absence of the SRY protein. So the SRY protein, if we have that present, then it's going to become a male. The absence of the testosterone causes the Wolffian duct to become degenerative. And in the uterus, the ovary and everything develops into the female sex organs. Because we have the presence of the anti-Müllerian hormone, it allows the Müllerian duct to become the fallopian tube and the upper part of the vagina. And with the male, on the other hand, we have once again 6 weeks after 6 weeks into fetal development, the internal reproductive organs have the potential to develop into the male structures if they're not into the female structures. And in the male structures, the SRY protein in a male embryo directs the medulla of the bipotential gonad to develop into the testes. The anti-Müllerian hormone from the testes causes the Müllerian ducts to disappear, as you can see here with the Wolffian duct. The testosterone from the testes converts Wolffian duct into a seminal vesicle, and the vas deferens and epididymitina DH controls the prostate development. So as you can see here, the internal male sex organs are reproduced, and then once that person's born, then they have all 3 of the sex organs that I had previously mentioned. So that is the end of this presentation of the determination of gender, and I hope you enjoyed it, and I included my references here, and thank you very much.